Have you ever considered why sprint champions don't also dominate the track in long-distance events? Of course, training and technique are important, but your genes play a significant role in determining athletic ability. From Archibald Garrett identifying the role of genes in disease in 1908 to the near completion of the human genome sequence in 2003, researchers have been captivated by our genetic code and what secrets are hidden within it. For many years now, the big question hasn't been whether our genetics are associated with athletic ability, but rather which genes are contributing to elite performance. One of the first of these being the discovery of the ACTN3 gene and its influence on skeletal muscle. Welcome to the genetic basis of stuff and things. The human body contains three main types of muscle: smooth, cardiac, and skeletal. Unlike cardiac and smooth muscle, skeletal muscle is under voluntary control and is broadly responsible for movement, posture, and strength. Structured in an extremely organized manner, skeletal muscle begins with several bundles of fibers called fasciculi. Each fascicle generally contains anywhere between 10 to 100 muscle fibers, depending on the demand for strength within that muscle, and consequently the type of fiber present. Whether it is type 1 slow twitch fibers ideal for endurance activities, or type 2 fast twitch fibers optimal for sprint and power activities. Moving further inside each of these muscle fibers itself, we find a series of cylindrical organelles known as myofibrils. Within these are repeating segments of microfilaments called the sarcomere. The sarcomere is the functional unit of skeletal muscle fibers. Composed of overlapping thin actin filaments and thick myosin filaments, the structure is bordered by two Z-lines. Within these Z-lines are various actin-associated proteins which cross-link the actin filaments, ensuring appropriate alignment with myosin and coordination of contraction across neighbouring sarcomeres. These proteins include alpha-actinin-2 and alpha-actinin-3. When the muscle contracts, the Z-lines are pulled closer together by the force of the myosin heads, sliding the microfilaments past one another. This shortening of the sarcomere produces the necessary force for muscle contraction. In 1998, the field of sports genetics was revolutionized with the discovery of the polymorphism in the angiotensin-converting enzyme ACE, which corresponded to enhanced sporting performance. Following this, in 2003, a collaboration of authors from Australia and the United States identified the role of the ACTN3 gene in sprint and power athletes. The ACTN3 gene encodes the alpha-actinin-3 protein, which, as mentioned before, is a key structural component of the Z-line in the sarcomere. Approximately 35 nanometers in length, alpha-actinin-3 forms an anti-parallel homodimer, with each subunit comprising of a central rod, two C-terminus EF hand domains, and an N-terminus actin-binding domain, which anchors the actin microfilaments to the Z-line. Initially, the ACTN3 gene was studied for its hypothesized relation to conditions such as muscular dystrophy. However, in 1999, researchers found that the alpha-actinin-3 deficiency was common within the general healthy population. This suggested that the deficiency is not associated with abnormal neuromuscular phenotypes. Through genotyping technologies, they noted the alpha-actinin-3 deficiency resulted from homozygosity for a nonsense mutation caused by transversion, converting an arginine to a stop codon at the 577th amino acid position. This prevents the complete translation of the coding sequence, causing a defect in the C-terminal domain, which ultimately results in a truncated protein that is rapidly degraded. Consequently, individuals who are homozygous for the nonsense allele do not produce detectable levels of alpha-actinin-3 in their muscles. Let's refer to the deficient allele as X and the wild-type allele as R. You may be wondering now, how does the sarcomere work without it? Luckily, in humans, there are two genes which encode skeletal muscle alpha-actinins, ACTN2 and ACTN3. Although they are very similar in function, ACTN2 is expressed in all muscle fiber types, whereas ACTN3 is restricted to type 2 fast-twitch muscle fibers, which are used for short but powerful bursts of energy. Considering this, researchers endeavoured to identify how alpha-actinin-3 deficiency phenotypically affected an individual. Using standard polymerase chain reaction techniques, they genotyped the DNA of 301 elite athletes categorized as either sprint and power or endurance. These individuals were compared to a subset of 436 control participants. Upon analysis of genotype and allele frequency, they found that sprint and power athletes had a much lower frequency of the XX genotype compared to the control and endurance groups. In fact, sprint and power athletes exhibited a notably higher frequency of both the homozygous RR and heterozygous RX functional alpha-actinin-3 genotypes. This led them to the conclusion that the ACTN3 577R allele provides an advantage for power and sprint ability and is a formative factor in determining the efficiency of type 2 muscle fibers. This conclusion supported prior phenotypical performance studies by researchers at the University of Antwerp, which demonstrated performance of elite decathletes in 100-meter sprint negatively correlated to performance in the 1,500-meter race. This once again demonstrates the critical role of alpha-actinin-3 in the explosive ability of type 2 fibers. Since the initial discovery of this relationship, various other studies have identified additional phenotypic features dependent on the ACTN3 gene. Denoting it as more than just a gene for speed, researchers also found its presence to reduce acute muscle damage, suggesting its protective role over the sarcomere. Overall, identification of specific genes and their correlation to enhanced athletic ability is pivotal to advancements within the sporting industry. By understanding an individual's genetic composition, training can be carefully tailored to their assets, whether that be speed, endurance, or strength. Such a personalised approach not only enhances the effectiveness of training, but also allows athletes to attain their greatest potential, ultimately improving performance outcomes in competitive sport.